Great. Well, welcome everyone. We are very excited today. Thank you for joining this webinar hosted by Thomson, partnered by Bionova, INFORS, Scarab Genomics. I'm Sam Ellis, the CEO of Thomson Instrument Company, and I'd like to introduce our partners, and everything will be recorded. Our lead speaker today, Fabio Fonseca, PhD, Director of pDNA Process Development and Interim Co-Site Head for Bionova. Myself, Sam Ellis, Marco Neri, the Director of West Coast Sales and Service for INFORS, Ann Stover, the Director of Marketing and Business Development for Scarab Genomics. And we will have a Q&A at the end of the seminar. Please type in your questions into the Q&A sections. And with our excitement, we're going to hand this over to Fabio to show some amazing slides.
Proceed slide, please. Hi. Thank you for everybody for being here and joining us today. Thank you for our partners, INFORS, Scarab Genomics, and Thomson. My name is Fabio Fonseca. I'm the current Senior Director of Plasmid Process Development here at the BNS. And today, I'm very happy to share some of our data and how BNS look like and what we are offering. So the title of our talk today is pDNA 2.0, introducing Bionova first-in-class manufacturing platform. Next. So I will start giving a little brief background who we are and how BNS is structured. We are part of Asahi Kasei, which is a titan company which has these global footprints and it's verticalized for bioprocessing solutions. So Asahi Kasei back Bionova Scientific, and this is the footprint around the globe. It's well. We can say that Asahi Kasei has a huge portfolio and including construction, biomanufacturing. It's a huge portfolio. But next slide. And one of the portfolio that I would like to share from Asahi Kasei, it's the bioprocessing portfolio. They are involved in purification solutions, microfilters, fluid management, and also biosafety and analytical testing. So that relationship to quality. And also the biologics and manufacturing. And we are part of these bioprocessing portfolio. Our site in Fremont, which is focusing on biologics, proteins and antibodies in Fremont, California, and here where we have GMP manufacturing of plasmid DNA and lentivirus. Next. Here it's a little bit of the overview of our facility here in Houston. We are small but mighty, and I like to say that we have a lean and mean technical group over here. We are designed to be our B4 building, this one on the left that you see. We are a GMP multimodal manufacturing facility sitting right here in the Woodlands, and we are 10,000 square feet GMP facility, up to 5-liter microbial fermentation, and we have a capability for manufacturing of clinical lentivirus. We offer end-to-end process development solutions, upstream and downstream, as well analytical development capabilities. And our facility is also set up as a multimodal facility to manufacture EV, LNPs, and peptides. And then we have another greenfield plan to have a commercial scale pDNA on a 70,000 square feet where the land was purchased and all the designing had been built for the building. It would be a 70,000 square feet GMP facility with a commercial scale of 200 liters and commercial LLV capacity. Next. Now I would like to switch gears a little bit and talk about the CDMO market. The CDMO market it's fiercely competitive, and as you can see here in the left, many companies are playing in the same arena. And in order to be successful, we need to differentiate it. And how do we differentiate it? And that's what BNS is focused here, in having that differentiation, having differentiators, and set apart from traditional CDMO and CMO business. The whole market in biologics is around projected to be around 50 billion by 2030. And specifically talking, the current market of pDNA, it's around $600 million with a projected expansion to 2 billion by 2030. And we are investing and being laser focused to play in this market and in this arena. Next.
So as of right now, I would like to paint a picture on the current autologous cell therapy. There is a trend, and you guys can track that. You guys can see there is an increase in clinical trial activity. There is a growing global interest in North America dominant and Asian and Pacific that are emerging. So it's very important for CDMOs and CMOs to be flexible and have the EU and as well FDA capabilities of manufacturing. The challenge here is the rising manufacturing complexity and cost as the systems become more complex with new technologies and as well impact of tariffs, for example. And then you see products coming from Ireland, from Eli Lilly with a 25% increase in cost, or products coming from Sweden with anything between 25 and 35% of increase. And also the lack of a point of care hospitals to administer these cell therapies, right? And with that, there is a new opportunity. With trend and challenge, it raises opportunity. And that is what BNS is being focused on, in new tech stacks that and be different than the version 1.0. And we are developing a version, a novel tech stack 2.0. And that goes back to the title of this talk, pDNA 2.0. And this 2.0 will be focusing quality, yield, and efficiency. And one of the things that I would call as a problem statement would be for lentiviral vectors and as well AAV, mobilization of a host of bacterial genes, including antibiotic-resistant genes, into the viral vector. This is a major concern, and BNS is striving to solve for that. Next. And this comes to insertion sequence elements contamination. What we have that these insertion sequences, they can transpose into plasmid regions, literally jumping into your plasmid. And these will lead to gene inactivation, alter expression, genomic plasticity, evolution, and antibiotic resistance spread when when this insertion sequence is part of a composite transposon. Here on the top left, you can see the IS element, which is the simplest of all transposons. It's a transposase gene. It's only one gene, and it's flanked by inverted terminal repeats that you can have basically two molecular mechanisms. One literally jumping through, and the other one cut and incision. And just underneath, you see how antibiotic resistance could be carried over to your viral vectors when these insertion sequences are part of a composite transposon. And then the benefits of having a IS-free plasmid, genetic stability, safety, regulatory compliance, because that's just a matter of time for the FDA to come with a new regulation and more stringent regulation for safety and quality and manufacturing efficiency. And with that, we go back to our switch from a 1.0 to a 2.0 novel stack, where we have BNS producing insertion sequence-free manufacturing, and this is possible because of one of our partners, which is Scarab Genomics, driving quality, yield, and efficiency. Next. So points of differentiation. These points, again, points of differentiation of our CDMO and CMO, that it's more, and you guys can see here, risk-based quality systems, proactive program management, and manufacturing excellence. And you guys are going to say, well, Fabio, any CDMO and CDMO has that as a standard, really, but it's a matter of adoption, truly adoption, and become part of the DNA, and that is culture, and that's what we are trying to do here at BNS. Well-designed, robust, and qualified process, Six Sigma lean approach, automation, data capture and integrity, and risk-assessed decisions. All decisions here at BNS are done through risk assessment and also data-driven, statistical data-driven decision. Flexible assets, like the trends in the market, single-use disposable equipment and closed system. Process automation, unidirectional flows of people and materials inside the facility. And well-designed process control based data. And then with that, we can have a agile operation model, clinical path resources, standard documentation, and technology-based differentiator. And one of the technology-based differentiator that will enable modeling, it's the digital twin process that we are going to talk about a little bit further in this talk. Autonomous system and unique bioprocess equipment. And we also invest in robust and well-qualified high-yield process. Next. Right. One of these differentiators is one of our partners that is called Cyanex. Cyanex has a unique 5 plasmid system for production of lentivirus. And one of those is the Cyanex T1, which solves for off-the-shelf speed and offers a 10x potency on T cells. This is the 5th plasmid on the lenti system that as you can see here on the graph, you have a 10-fold increase in potency, and then you can treat 10 patients with lower doses instead of 1. And these have been adopted heavily through academia, biopharma, and CDMO and CRO business right now. And BNS, we are the exclusive manufacturer of Cyanex plasmids. Next. Another differentiated, so BNS is differentiated itself on supply of plasmids to CDMOs. On the left, you have incumbent CDMOs with a price point of $400 per milligram. But the quality they have the insertion sequence contamination. And there are off-the-shelf solutions, IAV Lenti and AAV, which is third generation system. On the flip side of the coin, you have Bionova Scientific with a lower price, agile, and higher quality at $300 per milligram, a higher quality with Scarab Genomics and IS-free plasmid manufacturing. And here at BNS, the clients never have to pay any royalties for these productions. And we are able to achieve through genetic engineering of this cell line very low levels of endotoxin. Even at the PD environment, we are about 0.6 endotoxin units per milligram. Again, we are exclusive supplier and manufacturer of Cyanax T1. And we have off-the-shelf solutions for Gen 3 and Gen 4. And in the future, in the near future, we will have also off-the-shelf solutions for AAV. And we also offer custom plasmid design. Next. And then talking about flexibility and risk assessment. So here we designed a 10,000 square foot facility that is flexible and it's multimodal with complete closed systems, which leads us to capabilities of producing many GMP products, pDNA, Lenti, EVs, LNPs, and peptides. And with risk assessment and putting all the team together, thinking about that, we are able to degrade the classification to class D. And through isolators and closed systems, we can manufacture safer products. And with that, because BNS has in mind also the concept of sustainability. And with that, we can reduce trash, literally, and while solving savings on our operation, reducing gowning costs and the amount of trash that it generated, and also on EM. And all that by degrading and having a very smart process through, of course, risk assessment, we were able to achieve that here at BNS. Next. And here is the summary. As of right now, we have the highest quality and one of the safest plasmid DNA in the market. Best-in-class tech stack, true partnership. We have Cyanax, we have Thomson, and we have Scarab Genomics, and also for the 5-gram scale, we purchased the Sativa Flex Factory, single-use system for 5-gram scale. We have a reliable reproducible process through ultra huge and plasmid plus system from Thomson. We have off-the-shelf solutions for the for these 5 system Cyanax plasmid, T1, VSVG, GAGPO, REV, and also the backbone for the GOI. We have advanced host tools, Scarab Clean Genome Cell Line, E. coli. Flexible access, Sativa Flex Factory, single-use platform, and off-the-shelf solutions for all AAV serotypes, RepCap, helper plasmids, and GOI backbone. Next. So that's the summary of the services here in Houston, in the Udlands in Houston. Come to visit us. We are very happy to give you a facility tour. We have strain development. We have process development solutions and offerings, upstream and downstream, shake flask, feed batch, and platform customization. We have method development, upstream and downstream, as well qualification. We have formulation development. And our offerings on the RO material production, we have anything between 10 to 50 milligram. That would be the nominal scales. And of course, HQ and GMP manufactured for 500 milligrams and to come through Cytiva. We are fast tracking the development of the 5-gram scale and at HQ and GMP manufacturing. Next. Now let's see some data, right? And here I would like to kick off with our shake flask platform, first in class, one of a kind, and show some data here where the ultra yield system, the Plasmid Plus, outperforms two classically known high yield pDNA production media. And the example here, it's using DH5 alpha and the RapCap AAV RapCap 9. And as you can see, Plasmid Plus, which is in green, led to the highest OD of them all. And as you can see by titer, Plasmid Plus was able to enhance a higher pDNA titer based on mini-prep in comparison to CD Supreme and Circle Grow. Next. Plasmid Plus also outperforms two classically known high cell density promotion media. And in this case here, we are testing three different cell lines from Scarab Genomics, 1, 2, and 3. And as you can see, and this is cell line alone, very high ODs were able, we were able to reach using Plasmid Plus in comparison to coarse or Sturgeon. ODs on about 30. But there is a nuance. We are using here, apart from a great cell line, we are using the ultra yield system here from Thomson. Thomson not only offers a solution for OD and productivity, but Thomson also published and work with many Titan companies, helping them to achieve high production. And Thomson has a lot of white papers and publications through the website, and they actually guided us. And this system here, it's a 250 ml shake flask that Thomson gave us the know-how on how to fill. In this case, it was with 100 ml filling, and we were able to achieve these ODs. Next. So, and bringing back to that, we try to assess and to determine whether would it be the best filling for that 250 ml hyper flask. And as we can see here on the left, we have a filling with 50 ml, and on the right we have 100 ml. And you can see that depending on how you fill those flasks, of course, these were done at high RPM. That's another thing that should be mentioned. Thomson did a lot of work in the background, and then they have data supporting that 350 to 400 RPM yields more production, as Sam will be showing later. So we follow these, and these data demonstrate high ODs and higher production can be achieved faster when you fill with 50 ml. Next. And then, but of course, there is a little bit of evaporation that can be accounted in the system when you have a 50 ml fill, and these ODs and titers can be adjusted to the evaporation. But you can clearly see when you fill your 250 ml flask with 50 ml, you can produce higher titers and higher ODs faster than when you have a 100 ml fill. But either way, you're gonna get to the same production, but you can be faster, have a faster fermentation with 100 ml fill. Next. All right, but we are talking about a shake flask platform, and with that, everybody got to face the music, and we got to scale up. And then Thomson also offer shake flasks at a 2.5 liter capability, ultra huge shake flask at 2.5 liters. And the best recommendation from Thomson was to fill that with 500 ml. And here we show data and how much pellet and biomass we can get by using 3 different lentiviral system plasmids, VSVG, GAGPO, and RAV. And you can see that you can get from a 1 liter, right, basically 2 flasks, anything between almost 40 grams to 35 grams of cell pellet, depending on the plasmid. Next.
Fabio, we have 5 minutes left.
No, 19 minutes, because we had that I restarted my timer because we had that little technical problem, right? I'm at 19 minutes. So I'm gonna try to hurry up over here. So and these are the titers that we can get from these 1 liter 1 liter runs, 2.5 liter flasks. We can get around 2 milligram per gram of cell pellets. But there is one thing that I would like to highlight here is that if you are basing your process production, do not base it on mini prep, as you can get a 3 to 4-fold difference in the data. With mini prep, you would be overestimating your process. So the right way to do it's assess your productivity by giga prep. Next. And here, this is an example slide where we can highlight that every RCB and work cell bank that we make here at Bionova, we check our clones. This is a double digestion against the reference to show that we have that produce a cell line with the specific plasmid that we want to express, and then we confirm that by nanopore sequencing. Next. And then, but to really prove it, we produced the biomass, basically 13 flasks here with 500 ml, and to prove that we can have a shake flask platform for 500 milligram of the production. So we set up this biomass production and the cell patient generation and handle down for downstream. One thing that I would like to highlight here, that sometimes with these many flasks on a platform for in the GMP manufacturing flask, it's important to have these flasks synchronized. And as you can see here, we have some, these are the 13 points, we have some variation over here on the OD and of course on the acetate generation, because these flasks were not synchronized properly once it was all mediated, all performed by one operator. But despite of that, we were able to, from a 6.5 liter, to get almost 215 grams of cell paste. Next. The cell paste was put down on our lockdown downstream platform. We consist about 4 steps. TFF1, and this is our point of control for scale. We have a capture by AEX, and we have a polishing step by HIC, and followed by a final TFF where we can put it to final target concentration and final buffer formulation. Next. And here you can see, I'm very happy to say that our downstream process over here has a 30% loss, and we have a recovery of almost 73% of supercoil. And you can see by HPLC the quality of our pDNA coming from each one of these steps, and the final product over here at 90% supercoil. Next. And to that, we can have an overview and a summary from 6.5 liters of media. We can have almost 417 milligram of 91% supercoil plasmid, and then to adjust that to our downstream losses on 7.5 liters, we can deliver 517, almost 518 milligram of 91% supercoil period to pDNA. Next. And then here, I wanna highlight what are the difference, and why Scarab Genomics. It's such a beautiful cell line. Over 220 mutations and deletions, highly engineered to be highly produced, easy, high biomass, and high produce, and while being IS-free. And you can see that for each one of these plasmid tests, RapidCap, VSVG, doesn't matter, Scarab outperform every single one, DH5 alpha and DH10B. And you can see that you need on a downstream process, you need 8 to 13 liters of culture to achieve that 500 milligram. And if you look at how many flasks are going to be on the manufacturing floor, look at the delta between DH5 alpha and Scarab Genomics. So this is great. This is one of the best cell lines on the market right now to produce a high quality and high producer pDNA. Next. And we'll summarize and capture everything that I've been talking about. And we can see our scales 10, 15, and 50 milligram, HQ and GMP manufacturing 4.5 and 5 grams. And here you see the analytical, our 26, 280, right on the money against the reference between 1.8 OD. And then here we have beautifully 91% of supercoil on our final product. And on the right, you see that 14 sequences have been tested for presence of insertion sequences, and our BNS pDNA, which is here, the last load, completely absence of any one of these isoform. So this is a truly IS-free plasmid, first-in-class, as far as I know. This is the first-in-class IS-free pDNA manufacturer on the planet right now. Next. And this was very striking. This cell is engineered to produce less endotoxin, Scarab Genomics. And even at the PD environment, we are able to have 0.6 endotoxin units per milligram, which is absolutely fantastic. And of course, these will translate to our clean rooms. And just to point out that on our COV, it's less than 10, and we got 0.6 of endotoxin at the PD environment. Next. And this is our roadmap, our innovation roadmap, how BNS is looking to be in a 5-year period of time. Innovation added like platform process integration, in-house molecular library, and it's a bioprocess modeling centric on bioprocess model, reliable, reproducible, safety, autonomous, plasmid DNA manufacturing as well, LV, and many other gene and cell therapy delivery modalities, process intensification, all looking for a future state, 100% of a golden batch. How we're going to do this? Next. we're going to have digital twins. We're going to model our process. We are focusing over here in a commitment with digitalization and modeling, which is very sustainable too, because we can start measuring carbon footprint, and we can start measuring gas emissions, and have a more sustainable process and offer more sustainable process, because as Sahika say, it's a company that is committed with sustainability, so as BNS. And we will, with our partners, as our mother company, as Sahika say, and DataHow, we will have digital twins to support and to model our process. And we're starting with putting together upstream and analytical data together and start teaching, giving all that to machine learning systems. And next. That's how I'm going to end. And I want to thank you, everybody. I'm Fabio Fonseca, Senior Director of pDNA process development right here in the Woodlands. And one thing I want to say, our most important and our most valuable asset that we have is people, because without people, we don't have anything. And here at BNS, we take care of our people. And we managed to assemble a world-class team in cell and gene therapy with over 150 years of combined experience. And this is our greatest treasure. It's our people. And we are here, and you can see the picture of the team, because without them, we don't have anything. And this is our biggest asset. So thank you so much. I love our team. I love our processes here. And we came to stay, and we are different kinds of CDMO. We are flexible, we are agile, while producing the highest quality of products, and because we've got to take care of our patients. And one last thing, we are committed with sustainability, because we have one planet and 8.063 billion people. That's a lot of people to produce medications. So we got to be environmental consciential, what kind of processes we have, and what kind of waste we are being generating. And BNS is committed with that. Thank you so much. Now I hand off to our great Sam Ellis, the CEO of Thomson, one of our greatest partners, as well as Scarab Genomics, Cyanex, and of course, INFORS. Thank you so much.
Thank you, Fabio. Excellent. Well, I just wanted to introduce Thomson Instrument as a family-owned. It's my family. Just my sister, myself started it. My dad passed away about a month ago. Just want to say in honor of him, his science background at Columbia University, where he started, and science was always foremost and customer focused. And that's what we do. Everything's sourced here in America. Everything's made in Southern California. During summer or winter, you're invited to Southern California. We're one block from the beach here. So, if you need a beach walk in winter, just come let us know. Come summer, if it's hot, come let us know. Anyway, so we are here. We concentrate on cell growth, clarification, and purification. My background's a structural biochemist, and we're here making devices and innovation for our customers. So if you could next, please. So we look at things, if you don't have good pDNA, you can't transfect your HEC, you can't have good stable cell lines. So we look at that. We have our optimum growth flasks, our ultra yields, our new 6-well, our new 7-liter. We're consistently coming out with new products. We're working on new clarification products, and then new purification products for 2025 and 2026. So come, please interact with our team. Next. So if you look at it, we have over 48 patents, and we've come out with 200 products. And that's really been thanks to you, thanks to our customers. This has really been a collaboration and working together here. So looking forward to, again, new collaborations. Please reach out to our team if you're interested in other things that you'd be interested in doing. Next. So as I said, my grandfather was a chemist. My father was a chemist. They all used glass flasks, just like it was done in 1860. And, I think Abraham Lincoln summarizes it best. The honest truth, I wish I had Thomson flasks back in 1861. And you think about it, no one would use a rotary phone, but we all use an Apple or Android. But we have to look at it. We're looking at Scarab's vectors. We're looking at INFORS with speed. We're looking at things. We've got to look at our simple devices. It's the same thing with our flasks, our filtration, and our processes. Next. So the Ultra Yield flask, the Plasmid Plus, Ultra Yield was designed and developed with help with Genentech and Biosite Diagnostics, which is now Abbott or AbbVie. And Plasmid Plus, thanks to my friend Scott Leslie, Mark Knuth at Novartis. We did 1.5 years worth of work. They had 3 FTEs, and they had 1,000 transient transfection and gigas to get to. And we made a media that Fabio showed that could balance pH, use acetate, and grow to high OD with high quality transient transfection plasmids, with Fabio getting over 50 mgs per liter. Next. So again, we have something that's scalable from the 125 to the 2.5. The design was based on a reef in Seaside, where we took the angles of the reef so that, it gets high aeration and oxygen transfer rate, and with a large mouth. And next. So it's a combination. It's not just the Ultra Yield flask. It's not just the Plasmid Plus media. It's a combination of all of these factors together, which I'll go further in. So Plasmid Plus media can be used for scale up. Fabio's team used for, in fermenters with 2.2 liters to get 500 mgs. It can be used in flasks. It's scalable. It's simple. It's consistent. It can go to 22 to 24 hours. It comes stable, and it provides high DNA growth per liter. Next. So this is a paper from Oleg Brodsky with Pfizer and Pfizer La Jolla, where they showed just by changing the flask, they got a 610% increase on average. This is for proteins, but on average, what our product had done. Next. So here's more data that we worked with Pfizer. You can see on 1 through 4, an LB Nalgene nunc, by changing each one little parameter, how much better we could do. So you look at LB Ultra Yield, Plasmid Plus Nalgene nunc, which is just a standard glass or Nalgene nunc flask. Plasmid plus Ultra Yield gave a 530% increase in cell mass plus high quality DNA. Next. So we did a paper and white paper and poster with INFORS, and DNA quality is at #1 at our thinking. So we showed a 21-fold increase over LB, but an 88.5% supercoiled DNA. Next. So again, some of the things that we see, we can harvest longer, but RPM and speed, it needs 350 to 400. This is the process that it is definitely needed. We use a 1 to 5,000 antifoam, a Sigma Aldrich 204. Recommended, again, it's a 1 to 5 ratio, and then pick the right flask. So the 2.5 liter with 500 mil at 350 to 400 RPM is exactly what you need with the antifoam. Next. So again, we're looking at a 10-fold efficiency when using the Ultra Yield, the Plasmid Plus the vent cap. And that allows people to minimize their time, FTE time, and get results and give back to the lab in other things that they need to do. So I'd like to hand this off now. Come join us, Thomson Solutions at Work, and our new theme, which is Flask Us Anything, which obviously you can ask our team of experts anything, and we're here for you. So come visit us at PEGS. Come visit us also at ACGT. And Fabio will also be there, right, Fabio? So just to ask him other questions. And thank you. I'm going to hand this over to Marco.
BNS, just as a follow-up, BNS will be at the ISCT and also at the ASGCT, both in New Orleans, back to back.
Vic, what is our booth number? Does anyone have it?
723.
723. Great. All right, Marco. Thank you.
Thanks, Sam. That was great. I want to introduce myself as Marco Neri. I am the Director of West Coast Sales and Service for INFORS USA, which is the US subsidiary of INFORS HT. My mini presentation will be on confidence in bioprocessing, proven technologies you can count on. So just a little background, next slide, please. A little background on our company. INFORS was founded in 1965 in Basel, Switzerland. This year, we are celebrating 60 years of global expertise in bioprocess technology. So from our first shaker to today's trusted systems, we evolve with your needs. I really wanted to highlight that we are family owned. We're not a big conglomerate. We don't sell consumables. We are strictly incubator shakers, bioreactors, and fermenters. So capital equipment, hardware. This gives us tremendous flexibility to support our existing equipment on the field. We really do put the customer first. We put the customer at the core of our business. And our mission is to deliver smart, dependable tools that simplify bioprocess workflows. It wouldn't be a surprise if you went to an academic lab or a biotech lab and see our incubator shakers 10, 20 years old. That would not be a surprise. We really do stand by our equipment, and we're here for the longevity of the equipment as well. We're trusted by scientists worldwide to support critical research and development. Can we get to the next slide, please? So when Fabio's group initially came to me, they needed three main things. First was proven reliability. They needed to count on long-lasting performance, built for nonstop use. They were shaking on the higher end of things, so they needed something that can shake their cells 350 to 400 RPM with pretty fairly heavy loads, cultures and flasks totaling 19 kilograms or almost 40 pounds in each tray. They needed consistent temperature throughout the cabinet, even though they had exothermic reactions. So something that would keep temperature at all times. Second, they needed something flexible for their research. They were experimenting with different flasks. They were using Erlenmeyer's initially. They were using 50 ml conical vials initially as well too. And they finalized with the Thomson 250s and the 500 ml flasks. They also wanted something that was user-friendly, so they can pull out the trays and load them any way they wanted to. And they wanted a low working height and outward opening doors, so that could handle the 4-foot scientist or even the 7-foot scientist. We all know how that is. And finally, they needed to be possibly ready for qualification. So being able to put their system on the BMS or the building management system and possibly get ready for GMP. So as you can see, a lot of our equipment is made to order. We have different configurations for insect, microbes, yeast, mammalian cell cultures such as CHO or HEC. We do have a platform for 96 deep well plates with a 3 millimeter throw, and that is actually the diameter of the shaking orbital that can reach speeds up to 1,000 RPM. Our units come pre-stacked when they're ordered. If you were interested in only one stack and wanted to make a triple stack in the future, it's not a problem to, it's modular stacking, it wouldn't be a problem to do that. And then if we can go to the last slide, I know we've been talking about shakers and scaling up in shakers, but I just wanted a quick overview of our product portfolio. Again, we do have the shakers, we also do have parallel benchtop bioreactors. We have single vessel bioreactors all the way to the 10-liter working volume. And then when you go to scale up and tech transfer, we have options for stainless steel from the 10-liter working volume all the way to 1,000 liters plus. I thought I'd throw it out there. Can you get to the last slide? Let's continue the conversation. I appreciate everyone's time here. I'd like to introduce Ann Stover. She's Director of Marketing and Business Development for Scarab Genomics.
Thanks, Marco. And thanks to all of our attendees for joining us today as well. So we are the makers of Clean Genome E. coli, which Fabio talked about during his presentation, and which Bionova used to develop their pDNA platform. And as Fabio indicated, our Clean Genome E. coli strains are safer and more genetically stable than traditional E. coli strains. And they're also reliably production ready. So they're not only useful for small lab experiments, but they're already optimized and robust enough for large scale production. Our strains have been engineered to deliver higher yields and cleaner products. And whether you're working with proteins, peptides, nucleic acids, or even single chain antibodies, we likely have a clean genome strain in our library that will work well for your application. In addition to our strains, Scarab has also developed a proprietary continuous fermentation technology called cFlow. And when our clean genome strains are combined with cFlow technology, they enable continuous fermentation. So I'll talk more about that in a second, but first I just want to give you a brief look into how we got started. Can you go to the next slide, Vic? So we were founded by Dr. Fred Blattner, who's pictured here on this slide. Dr. Blattner is a pioneer in the field of E. coli. Working out of his lab at the University of Wisconsin in Madison, Dr. Blattner and his team were the first to fully sequence the E. coli genome. And later they developed the multiple deletion series E. coli strains, which we now refer to as Clean Genome. So Scarab Genomics was established to commercialize these strains and take them from the lab into scalable bioproduction. Today, we're still based in Madison, Wisconsin in the US, and our mission is to continue to leverage Dr. Blattner's work by delivering safer, more stable cell factories that help optimize production processes. So let's take a look at the core of our technology, which is Clean Genome. Clean Genome started with the parent strain MG1655, and from there we removed about 20% of the genome, including all mobile DNA sequences and phages. This reduction enhances growth efficiency and makes the strains much more stable, both genetically and physiologically, which ensures reliable performance over time and across scales, and produces consistently cleaner products. We're also continuously engineering new versions of our clean genome strains with additional targeted gene knockouts, metabolic optimizations, and other custom modifications. So while only a handful of our strains are listed publicly for licensing, we actually have a very extensive library of strains that we've developed internally, which gives us a lot of flexibility in terms of matching a wide variety of production needs. Additionally, our technology is all IP protected, which gives our customers the security of a proprietary platform. As I mentioned earlier, we've also developed a continuous fermentation technology called cFlow, and this was designed exclusively for use with Clean Genome E. coli, which is really the secret behind its success. Clean Genome provides the genetic stability needed to maintain that steady state fermentation over weeks or even months. It's a very compact system requiring a small footprint of only about 200 square feet. And because of the stability of our strains combined with the design of cFlow, process development is faster, which in turn helps accelerate time to market. So let's look at a quick example. This graph shows production of the vaccine carrier protein CRIM197 in clean genome, which is shown in blue across the top, versus 4 traditional E. coli strains, which are the other colors down in the bottom left. And so what you can see here is over the course of a 35-day fermentation, clean genome maintained a high level of production of CRIM, averaging about 4.5 grams per liter. By comparison, the B strains and MG1655 only maintained adequate production for a couple of days. We did deep sequencing at both the start and end of the production run, and the results were really clear. What we found was that clean genome had only a very few number of mutations and no plasmid changes, whereas the sequencing done on the BL21 strains showed such massive rearrangements and degradation by the end of production that it could not be assembled. So this really highlights one of the key advantages of our platform, which is long-term genetic and expression stability that supports consistent, scalable bioproduction. So that was a very high-level overview, but if anything I've mentioned resonates with your production needs, we'd love to continue the conversation. I have my email address listed here, as well as for Charles Landry, our CSO, who's on the call as well. And you can either contact one of us, go to our website, scarabgenomics.com, or just scan the QR code here and someone will reach out to you. So I think with that, I'm going to pass it back to Sam.
Great. Thank you so much, Anne and Marco and Fabio. Questions and answers. Just, let me just go through. First one, Fabio, can you comment on how the improved production affects the time to harvest? Do you recommend waiting longer to allow more production or harvest earlier at some peak? Can you go through that?
Yes, each system, it depends what you're manufacturing. In the case of our pDNA, we learn here at BNS that pDNA production after biomass. So first you have that burst of biomass increase. In a shake flask, within 5 to 6 hours, all the glycerol had been consumed, and then it starts metabolizing acetate. This system is very interesting because it kind of like self-titrates. And here at BNS, we learn that plasmids are producing during low glycolytic, in a low glycolytic phase. So after all the glycerol or glucose, in the case of Scarab like is glucose as number 1 substrate. After that, it metabolizes the acetate, and then you leave for another few hours, and then you harvest. And we show that you can sometimes have a production within even 15 or 18 hours. In the case of the pDNA, I rather have, and this is what we are thinking and we are looking, have a shorter fermentation times as you're going to have a turnover. Right, your cells are going to be dying. That with death, you produce cell death factors, and then you can lead to cell death. Of course, you're going to damage your product. So we are focusing on shorter fermentation times, anything between 18, 20 hours, maybe sometimes 22, and fermentation for sure in the fermenter less than 24 hours. So there is a balance. You should measure your metabolites, and that will dictate your harvest time. But for sure, using the ultra yield system, you can have definitely shorter harvest times while maximizing your production. We actually did mini preps time courses, and there is a sweet spot. Anything between 18, 20 hours would be appropriate to harvest, sometimes 15. It depends also on the plasmid that you are trying to manufacture.
Yeah, and Fabio, we have another question. With Plasmid Plus and Ultra Yields, are you showing that you have a nutrient depletion? And is that really true that acetate goes down? Is that really a function?
It's a beautiful thing. Over and over we see this. and we see these even at the fermenter. That's what we mean by low glycolytic. Acetate is down, and all your carbon source is down, and then you have you basically stop growth, and then everything it's shifted to plasmid production, and then we harvest. Yes, it happened every single time, but there are nuances depending on the plasmid that you are doing. But overall, your acetate it's really low in the flask. But we've seen some flasks in some constructs. Depends on the producer cell line, that at harvest time you have around 5 on the acetate. So it depends, but overall the trend acetate is down, and that's the beauty. It's like a self-titrated system.
Yeah, so pH is stable, Fabio, you're saying that?
Beautifully. You see sometimes depends on the growth, because some producer cell lines they grow a lot. You see that dip on the pH, but after and then after the carbon source is gone, you start the main ones, the glycerol or your glucose, you start degrading metabolizing the acetate, and therefore you see that rise in pH, but throughout the system, it doesn't stay low or very high. It's very consistent, the pH. There is some variation, but very consistent.
Yeah, so your cells don't require any special medium besides the Plasmid Plus. So you just stick the Plasmid Plus in your shake flask. There's no other special media. It's that high speed 350, 400 RPM Plasmid Plus plus an ultra yield and a vent cap. It's that simple?
It's that simple, that easy. Of course, we have a great incubator from our partner here, INFORS, great incubator triple stack. That's all that we need. But we see some evaporation at a 50 ml filling, but at this scale, nothing to worry about it. That's all you need. Plasmid Plus, ultra filter, the ultra yield system, you're good to go.
And then we have another question for Ann and Fabio. Have you compared these strains to NEB, STBL, NEB10, STBL3 for DNA yields and stability, Ann and Fabio, or whoever from Scarab?
Not that I know of. Charles, do you know if we've compared to those strains?
We have not.
Yeah, we haven't. I heard that these strains are also great as producers, but I'm not sure that if they are IS free, because here we are looking not only for high yields and production, but we are laser focused on safety and quality, and that's why. As far as I know, the only IS-free platform that we have is through Scarab Genomics. So never tested. I would love it to see how we perform, but unless if they are IS-free. If they are not, I just use it commonly now, the H5 alpha and the H10B to have some controls, but we're laser focused on IS-free cell lines.
Yes, thank you. And we had a question for larger plasmids, 15 kb. I'll let you know my experience. And Tara's here from Advancion. We did some backmids, plasmids that were 100 kb, and we got a 4 to 5 fold increase by using Plasmid Plus ultra yields consistently with the high speed. Again, those backmids are huge. Above 15, we were at 100 kb. So, and then another question from Danica. I know, Fabio, you did 2 liters. We have had customers do up to 10 liters. She said, how does the fermenter perform with Plasmid Plus?
Beautiful question. We are we are here, and I'm pleased to say we will have these locked down. One thing I want to say, even Sam was showing his data, we have a 91% supercoil going through downstream, a little higher than Sam on his slide, because we have a beautiful downstream platform. But I can share that for the 500 milligram, what we are seeing here, it's less than 5 liters. To be precise, 4.8 liter fermentation with Plasmid plus, and using Plasmid plus glucose as feed for the Scarab, that's enough to have 500 milligram. And one other news that I want to say, by right now, all our cell lines, I just received the email, all our cell lines have been sequenced by NGS, and now we have the data, so this I can't stop smiling, and we have that sequence on our product. So this is great. Yes, 4.8 liters with Plasmid Plus, and using Plasmid Plus plus glucose on your feed will deliver you 500 milligram if you have a tight downstream like BNS does.
Wow. Well, that's pretty amazing, Fabio. Someone asked, are you avoiding LTR, ITR mutations using DH5 alphas?
We're not, again, the DH5 alpha, we only
You're going to
use it-
scarabs because you can reduce with that. You can reduce. The clean genome has helped you guys in a lot of with the plasma deletion that really has given you that extra boost, right, Fabio?
100%, and also this is what we like to say with our partner Scarab Genomics. This is our 1.0 version of the process, but there is more to come. These cells are being further engineered for situations like that, and actually Scarab Genomics already has cell lines that can deal with harping, secondary structures, and ITRs, and hard to produce plasmids. But we are going to go a little further in these particular clays and start incorporating some of these deletions and mutations to facilitate ITRs and etc. Right now, we are focusing on lentivirus, but that will be more on the AAV. Yes.
Great. Well, thank you, everyone. Please reach out to our partners here, to whether it be your Thomson partner, Bionova partner for production, and for Clean Genome or Marco. We'd like to thank everyone, and contact us in the future. There's going to be some more developments as the team works together. And I just want to thank Fabio, Ann, and Marco for a great time. And it really wasn't being able to be made without Vic in the background and the Thomson team. So I would like to thank them for our first webinar that we did. And thank you, everyone.